For centuries, the biological world seemed perfectly straightforward. If it was green and stayed put, it was a plant. If it moved around and ate things, it was an animal. This was the two-kingdom system introduced by Carl Linnaeus, which neatly divided all of nature into just two distinct halves. But then we invented the microscope. Looking closely at a single drop of pond water revealed a chaotic, invisible universe swarming with microscopic life that completely ignored Linnaeus's rules. Take the euglena. If you expose this microbe to sunlight, it photosynthesizes like a standard plant. But plunge it into darkness, and it begins hunting and consuming other microbes exactly like an animal. Or look at mushrooms and other fungi. They grow out of the ground like rooted plants, which is exactly where early scientists placed them. Yet they don't use sunlight at all. Instead, they feed on dead, decaying organic matter. Categorizing life by what it looks like from the outside clearly didn't work. To build an accurate map of nature, scientists realized they had to look deeper, organizing organisms by their fundamental microscopic cellular blueprints. In 1969, ecologist R.H. Whitaker proposed a solution, the five-kingdom classification system. This model sorted life based on hidden criteria: cell structure, body organization, nutrition, reproduction, and evolutionary history. At the foundation of this blueprint sits Kingdom Monera, single-celled prokaryotes completely lacking a membrane-bound nucleus to hold their DNA. Some of the oldest members of this group are the archaebacteria. They thrive in environments that would kill almost anything else, from boiling hot hydrothermal vents at the bottom of the ocean to the oxygen-starved stomachs of grazing cows. Contrast those extremophiles with eubacteria, or true bacteria. This massive group includes cyanobacteria, microbes equipped with chlorophyll, allowing them to harness sunlight and produce their own food through photosynthesis, much like a plant. Despite their lack of complex internal compartments, monerans are incredibly resilient. They form the rugged biological baseline from which all other, more complex kingdoms eventually evolved. That next evolutionary step brings us to Kingdom Protista. This marks the arrival of eukaryotes, the first single-celled organisms to house their genetic material securely inside a true membrane-bound nucleus. Protists display an incredible biological economy. Aquatic diatoms, for example, build indestructible walls out of silica. They essentially live inside microscopic glass houses that protect them while still letting in enough light to photosynthesize. But not all protists are so benign. Dinoflagellates are marine organisms propelled by two whip-like tails. When certain species multiply rapidly, they trigger toxic red tides that suffocate and kill massive numbers of fish. Moving on to dry land, we reach Kingdom Fungi. Unlike plants, fungi are multicellular heterotrophs, meaning they cannot make their own food. Their cells are reinforced by rigid walls made of chitin, a tough material completely different from plant cellulose. Because they can't rely on the sun, fungi have to forage. They do this by secreting digestive enzymes into their surroundings and absorbing the broken-down nutrients through massive, sprawling underground networks called mycelium. As we can see on the left side of this graphic, fungi secure nutrition in three ways. They can be saprophytic, breaking down decaying matter, parasitic, causing diseases in plants and animals, or they form mutually beneficial symbiotic relationships. There is even a peculiar class known as deuteromycetes, or imperfect fungi. They earn this title because scientists have only ever observed their asexual or vegetative phases. Their methods of sexual reproduction remain a total mystery. Together, protists and fungi illustrate how that initial, microscopic eukaryotic blueprint rapidly diversified. It branched into highly specialized strategies, yielding both complex aquatic predators and highly efficient terrestrial recyclers. Whitaker's five-kingdom system elegantly organized cellular life. Yet, as our microscopes grew even more powerful, we found a glitching, uncharted corner of the biological map. We discovered entities that defy the strict criteria for what is considered living. Consider viruses. Outside a cell, they are entirely inert, crystalline structures. They possess no cellular machinery of their own and remain utterly lifeless until they succeed in locking on to a living host cell and hijacking its internal systems to replicate. Then there are viroids, which cause diseases in plants. They are significantly smaller than viruses and consist only of naked, free-floating RNA, entirely lacking the protective protein coat that a typical virus carries. Prions break the rules even further. They contain absolutely no genetic material at all. They are simply rogue, infectious proteins capable of degrading nucleic acids and causing severe, often fatal neurological disorders in mammals. This diagram illustrates another anomaly on the right, lichens. Look at this cross-section showing the symbiotic masterclass. Algae produce food through photosynthesis, while the surrounding fungi provide shelter and absorb water. Because they are so deeply interconnected with their environment, lichens act as highly sensitive indicators, unable to grow anywhere near pollution. Biological classification is an ever-evolving map. Nature rarely confines itself to our rigid categories, proving that as long as our investigative technology advances, we will always find strange, rule-breaking anomalies hidden in plain sight.